Colócale mejor a todos esta
¿Sí?
y yo tomo
Una mineral.
gracias, agua.
Why do we have two seats there? Why do you need two seats there?
Just to have 4 and 4, but
Ah, okay. So then we can use that one. No, I said we want to spend it for John's ideas. I respect them.
No, querías esta, ¿no?
todos de la
Ah, you want the same color. Okay, John. You see. It's actually a plan. What's the
It's like less cold than before, is it?
Yes, I think so, but it's supposed to look at the people are moving it. But relax. Do you need anything on top? We're going to take it.
That's a good sign.
Aqui?
Thank you, Mark.
Very well. Anything else?
No, me trouble.
Let's say for the
that okay? I think so. Do you want me to disinfect it?
Dames en heren, geachte collega's, ik heet u van harte welkom op de openbare doctoraatsverdediging van John Freddy Morales Tellez en verklaar de zitting voor geopend. De heer John Freddy Morales Tellez is Master of Biomedical Engineering van de KU Leuven en heeft aan de faculteit een proefschrift voorgelegd handelend over Respiratory Sinus Arrhythmia Estimation, Closing the Gap Between Research and Applications, wat in het Nederlands vertaald wordt als schatting van de respiratoire sinusarrhythmie, de kloof tussen onderzoek en toepassingen dichten. En dit tot het verkrijgen van de graad van dokter in de ingenieurswetenschappen. Hij zal het proefschrift nu verdedigen en ik vraag aan de kandidaat een uiteenzetting van ongeveer 45 minuten te geven over zijn werk. Daarna zal er gelegenheid zijn tot het maken van opmerkingen en het stellen van vragen. Dear candidate, I invite you to present your work in 45 minutes. The floor is yours.
Thank you. Oh, just put it there. Good afternoon, everyone. Thank you for being here today, either in person or online for the people in Colombia, Mexico, and Argentina. I will say something in Spanish. Gracias por estar acá. A la gente que está acá en persona, también los que están en México, en Colombia y en Argentina en este momento. Voy a hacer una presentación de 45 minutos, va a ser en inglés. Traté de hacer los slides comprensibles y pues disfruten y ya al final diré algunas palabras para que se queden conectados. Voy a cambiar inglés en este momento para hacer la presentación. So, yeah, today I want to talk about my PhD research, basically the things that I was doing for 4 years here in Belgium. And I studied methods for the quantification of the respiratory sinus arrhythmia. And I developed some tools to try to better understand these methods and to try to bring them closer to some applications. And this is done studying the electrical activity of the heart. So it's normal to start talking about the heart. You can see an MRI scan here of the movement of the heart. And it has a function which is to pump blood through the body to bring nutrients and oxygen to the cells. But to achieve this, there are some electrical circuits in the heart that coordinate all the activity of the muscle cells. In this part here, we have something called the sinoatrial node, and this group of cells coordinate the whole activity of the heart. We can record it with some electrodes attached to the chest, and we will see a wave, like an electrical wave, like this one here. And depending on the cells that activate at some moment, you will see different waves in this piece of signal here. Let's say that the most important for my studies was the R wave, this peak here, because it's an easy wave to detect in the ECG signal, and it allows to do some studies of the electrical activity of the heart. So as I mentioned before, the ECG signal can be recorded with a device. It can be recorded with 2 electrodes that are attached to the chest. And then you will have the same waves that I showed before, but one after each other. From this signal, you can detect the R peaks. And then you can also build a time series in which you plot or you put the intervals between consecutive R peaks over time. So you start with the first one, then the second one, and so on and so forth. And you build a time series like this one here. This is looks very simple, yet it provides a lot of information about the functioning of the human body. And the activity of the heart is modulated by different physiological factors and different physiological systems. Two of them, for example, are connections to the central nervous system, as well as some hormones. Specifically talking about the central nervous system, there are two different control mechanisms or two different branches that control the activity of the heart. One that tells the heart to relax, to slow down, and one that I have very activated right now that tells you that it's the moment to have energy and to like be ready because there is some I don't want to say this, but something happening that could be a danger. Then let's say that you are at home, you are standing, so your body is in this state here. So your heart is beating and you have this, with this rate here. You decide to go for a walk to the park, maybe to a forest here in Heverly, which is very nice. And at that moment, the running branch, let's say, of the autonomic nervous system activates a little bit. And some hormones also activate because now you are moving, so your body needs a little bit more of energy to keep the functioning of the body. At that moment, this gets a little bit shorter. The heartbeats occur faster, and then suddenly a dog appears and barks. So at that moment, you, of course, get super scared. So this activity here gets overactivated, and the time intervals between consecutive R peaks get shorter. Then you go home, you relax a little bit, you breathe, and the activity that tells your body to relax activates, and then after some time, everything goes back to what it was before going for this walk. So, as you can see, in this very simple example, the heart tries to adapt to things that are happening in the environment. The same occurs with different diseases and conditions. Like if you are stressed, the activity of the heart is going to be different compared to when you are relaxed. Also, if you have some diseases like sleep apnea, which is a problem in which people don't breathe, stop breathing during the night, we think that also this activity of the heart is different. So analyzing the tachogram and the heart rate variability allows us to evaluate the health condition of the people. So far, so good. We have the heart. I talked about the connections from the central nervous system to the heart and the hormones. But there is a very important modulator of the activity of the heart, which is the respiration. And it is natural to think that the two systems are coupled because, as you can see here, they interact a lot. And this is from a mechanical point of view, but also there are some sensors here of pressure and temperature and that also modulate the activity of the heart in synchrony with the respiration. Now, there are two different modulations of the activity, or two different effects of the respiration on the recordings of the electrocardiogram. So we go back to our figure now. And you can also record the respiration with a device. It can be, for example, a belt or two belts that you put around the chest or the stomach. And the signal will look like this. The first modulation that you can observe is in the ECG signal itself, in the electrocardiogram signal itself. So you will observe that the amplitudes of the heart peaks are not constant as it was before, but they change in synchrony with the respiration. The second type of modulation is the main subject of study of my PhD, which is the respiratory sinus arrhythmia. So if we take the intervals between consecutive heart peaks, we will see that during inhalation they get shorter, and during exhalation they get a little bit longer. And this phenomena is not very clear, like we don't know exactly the reason for it to occur. We also don't know exactly the physiological function of the respiratory sinus arrhythmia, but from research we think that it changes in different conditions and diseases. So it is important to quantify this phenomena because we can use it to evaluate the condition, the health condition of a person in an easy way. So basically, during inhalation, the heart accelerates a little bit. During exhalation, it slows down a little bit, and this is the effect of the respiratory sinus arrhythmia. Now I want to explain one type of analysis of the heart rate variability in which I focused a lot. But for this, I want you to think about paint, so of colors. Let's say that our heart rate variability signals are comparable to different colors. And you want to mix some primary colors, some more basic colors to obtain this. So basically, you need to put a little bit of red, a little bit of yellow, a little bit of blue, maybe some black, some white. And then when you mix them, you obtain the colors that you want to get. In the same way, the heart rate variability can be, or these signals can be obtained by decomposing them into different frequency components. So my frequency components are like different colors that I have to mix to obtain the original signal. So it's like I'm trying to see the same signal from two different points of view. When we analyze this spectrum here, then we can find that different activities of the body are reflected in different frequency bands. Like each activity has some specific colors, let's say. And the colors of the respiration are in this band here in the heart rate variability. And the standard to analyze the heart rate variability tells us that here we can see the activity that relaxes the body is reflected in this band, as well as the respiration. In this band, we see more activity, like the running activity that I was explained before, as well as some, a little bit also of the relaxing activity. And the standard to analyze this spectrum tell us that there should be like two peaks in these two bands. But this is not always the case. Sometimes you have a broader respiratory information here. Sometimes the respiration falls here or even here. So there is a need to understand or to have some more methods to quantify the respiratory sinus arrhythmia other than using these colors that I was explained before. And in my PhD, I investigated these methods. I studied these methods, and more like the current methods, and I developed some tools to try to compare them and to try to bring them closer to real applications. Just to summarize the challenges of the respiratory sinus arrhythmia, so we have a phenomenon called the respiratory sinus arrhythmia in which the heart accelerates and decelerates in synchrony with the respiration. From research, we know that it changes in different conditions and diseases. Here you can see some examples. However, the origin and function of the respiratory sinus arrhythmia is not completely clear yet. There are many methods for the quantification, but these ones are not standardized. And the studies are mostly done in controlled environments and healthy populations. So we need to improve our understanding of the methods. With all of this that I have told you so far in mind, this thesis had 4 objectives. First, to do a quantitative evaluation of the methods for the respiratory sinus arrhythmia quantifications, to study the methods in controlled and non-controlled environments and like in unhealthy conditions. Also to study the linear and possible nonlinear interactions between the cardiac and the respiratory systems, and to use the methods in different applications. So let's go to objective number 1. For this, I developed a model of the respiratory sinus arrhythmia. And at this point, I want you to think about coffee. So this is something that we grow a lot in Colombia, so that's why I... So yeah, this is the cycle of the coffee. This is the actual flower of the coffee, if you're curious about it. From the flower, then you get the grains, then you have to take them, you have to dry them, then you have to blend them, and you have a nice cup of coffee in your table, Colombian coffee. And the activity of the whole cycle is modulated by different things. One of them can be the weather. So the coffee likes rain a lot. So yeah, you need a lot of water for the coffee to grow well. And you have some other factors that can affect or that can improve or deteriorate the way in which the coffee is produced. So if you have more rain, you will have maybe more coffee, and so on and so forth, right? And then if you analyze this over time, you will see that if you know the cycles of the rain in the different months of the year, maybe you can also more or less have an idea of how much coffee you are going to have at some specific moment. The coupling or the relationship is not perfect. Maybe here the correlation is not perfect, and this is because you have the action of some other modulators in the, or some other factors in the, not only the rain, but some other things that change the coffee production. If you see the 2 time series, this is the respiration, these are the RR intervals. So in this sense, it's a similar activity, and you can do a simile here of the activity of these systems. So basically, we can think on the modulation on the rain as the respiratory activity. Other modulators can be the central nervous system and the hormones. All of this comes together to change the activity of the human body. This is reflected specifically in the heart and the lungs. And then our output can be the signal that we see, right? This is our coffee. And now let's go more abstract. So the respiration can be modeled as a signal. The other modulators can be also modeled with another signal. And then we have a system here that we can model. Well, in this case, it's the addition of these 2 signals together with a coefficient. This coefficient is going to tell me how strong the coupling is. So this is basically the model that we used here to compare the different methods. And the output that we will have are some pulses. Each pulse represents 1 heartbeat. And we used this model to compare different methods for the quantification of the respiratory sinus arrhythmia. So the idea was to take this signal here, the respiration, this signal here, and then combine them in some way with some mathematical tools to obtain an estimation of this coefficient here. The closer this estimation was to this, the real coupling, let's say, gave an indication of how good one estimator of the respiratory sinus arrhythmia was. This was the model, and we used it to compare different methods. I won't go into the details of all the methods because it would take 1 day. But I will explain 3 groups of methods that we used. The first one is the group of the colors that I explained before. So you have the heart rate variability, you decompose it in the different colors, and then you take the colors of the respiration, and you quantify how much of these colors are contained in our heart rate variability. The second group are non-parametric methods. So basically you take the information, in this case rain and coffee, you combine them together and you try to see how well correlated they are. The more correlated they are, the more coupled they should be. Remember, the rain would be like the respiration, the coffee would be like the heart rate variability. And then we have a group of parametric methods. Here, basically, we take one point in time and we go back to the past, and using the past information of the amount of rain, we try to see the amount of coffee that we will obtain. Again, this would be similar with the respiration, and it would be similar with the heart rate variability. And these are the results. Don't be scared. These are just errors. So the higher these are, the higher the errors. So the group of the colors, the group of the power spectrum has the highest errors, let's say. And yeah, this occurs because we consider specific frequency bands, and sometimes the respiration is actually broader. The second group were the non-parametric methods. They were better, but we found out that the parametric approaches had the best performance in our studies. So this was the main conclusion of this, one of the main conclusions of this work. And yeah, we produced a model of the cardiorespiratory coupling that is available online, and it allows to compare any method for the quantification of the respiratory sinus arrhythmia. Yeah, it is available online, and we found out that the methods based on subspace projections and cross entropy were better in this case to quantify the respiratory sinus arrhythmia, the changes of the respiratory sinus arrhythmia. So then we took these 2 methods and we compared them a little bit more. And the idea was to evaluate them in uncontrolled conditions and in unhealthy populations, as well as one important factor that needs to be considered that I will explain now, which is this one. So remember, we take the past information of the amount of rain to estimate the amount of coffee. But if we take part of the past, we get some estimation here that is a little bit out of the real value. Maybe if we go a little bit more to the past, we will get closer to the real value. But the question is, how much in the past do I have to see to be able to have a good estimation of the cardiorespiratory coupling. So this was the first factor that we investigated in this objective. And these are the results. Remember, this is subspace projections, this is cross entropy. These are the two parametric methods that we studied. And basically, we see that the model order, or the number of past information that we have to consider is actually not that important, but what is actually important is to always take the same model order for all the analysis that I do. That was the conclusion from this study, and this happens with both methods. The second factor that we considered here is the occurrence of irregular heartbeats. On the right, you can see a heart that is working normally. So you can see how the ECG looks very nice. The shape that I showed before holds. But sometimes the electrical activity of the heart is not working properly. This occurs for different diseases, or sometimes when people get older, this condition also appears. In some healthy people, you can also see this, but it's not very frequent, and it can be also normal. But the point is that the ECG signal looks completely different. So the question was if we could quantify the respiratory sinus arrhythmia using this type of signals, and up to which degree could we do it in a good way. And here are some results. So basically, we found out that even with a low number of irregular heartbeats, the quantification of the respiratory sinus arrhythmia was significantly deteriorated even after applying a correction of irregular heartbeats. The next factor that we wanted to check were delays between the signals. So the respiration can be recorded in different ways. The one that I explained before is called inductance plethysmography. So basically you put two belts, one in your chest, one in your stomach, and you can see how the respiration is. But you can also use the airflow in the nose, or the bioimpedance, in which you attach electrodes to your body and you analyze some electrical activity to extract the respiration. The thing is that each signal can be delayed differently with respect to the activity of the heart. So it was important to try to understand how the respiratory sinus arrhythmia estimations could be affected by this delay. These are the results. But again, in this case, the delays didn't have a significant effect on the quantification of the respiratory sinus arrhythmia. And the reason for this is that you are combining, like taking a lot of information of the signal from the past, and this gives some robustness to the methods. The last factor that we studied were phase changes between the signals. So we have observed that in some situations, the coupling between the cardiac and respiratory systems gets like is inverse. Like instead of increasing during inhalation, the heart rate decreases during inhalation, and vice versa. We wanted to see how this effect could deteriorate the quantification of the respiratory sinus arrhythmia. And it would be something like this. So in this part, instead of having increases and decreases as here, they are inverted. We simulated this scenario and we found out that this has a significant effect on the quantification of the respiratory sinus arrhythmia. And it is because these methods are linear and they are not able to overcome these problems in the signals. So with all these experiments, we produced a set of recommendations to try to use the respiratory sinus arrhythmia quantifications. This is also published for people to use it. And yeah, as explained before, the model order can be just fixed in a fixed value. 5 irregular heartbeats in a period of 5 minutes are already produce already significantly deteriorated quantifications. Both estimates were found to be rose to delays, and then this degree of change of phase is tolerated by the quantifications, by the estimations. Okay, now let's move to objective number 3. Here the idea was to see linear and possible nonlinear interactions between the cardiac and the respiratory systems, and here I will go back to the coffee. So maybe in some period you have some amount of rain and some amount of coffee. In the next period, the amount of rain increases and you have a linear increase in the amount of coffee. For the next one as well. But then you see that we have more rain, but it changes a little bit. It's not that linear anymore. And then more rain, but it doesn't change the amount of coffee that you have at some point. So we see that actually, in this case, the relationship is non-linear. And we think that quantifying this in the cardiorespiratory system can also provide important biomarkers and important information to evaluate some diseases and conditions. For this, we used, again, a kind of parametric method in which we try to build a regression in which samples of the heart rate variability are estimated using the past of the respiration. This is able to capture either only linear interactions or both linear and nonlinear interactions, and a mathematical function is used for this task. And with this, we produce two parameters. This one, which is the linear part of the linear estimator, and this one that is able to quantify both linear and nonlinear interactions between the cardiac and the respiratory systems. And we evaluated this during sleep, but I need to explain a little bit about sleep for you to understand. So normally when we go to bed, we go through different sleep stages. In the rapid eye movement is the lightest sleep stage. We still it's called a rapid eye movement because you have your eyes closed, but the pupil moves a little bit, and then you go deeper and deeper in deeper sleep stages. And during the night, you move through different sleep stages, and this is completely normal, and depending on the patterns that you show here, you may sleep better or worse. The idea here was to see the linear and nonlinear interactions during different sleep stages, to try to see how they changed in different sleep stages. And these are some of the results. Again, don't get scared. I will tell you the main message, which is that the nonlinear interactions, we didn't find any significant, that they were significant during the different sleep stages. So basically, in this case, the linear part of the interactions were dominant during different sleep stages. So with this, we presented a method able to detect the dominant form of coupling, and the linear part was found dominant in the dataset that we used for different sleep stages. Finally, for the last objective, this thesis has different contributions in different applications. And here first I want to talk about the ECG-derived respiration. So remember that one form of modulation that I explained before is in the amplitude of the R peaks in the ECG signal. So we can extract them, and we will see that these amplitudes will follow the respiration. This means that we can use the ECG to also estimate the respiratory signal. So in terms of applications, well, I used the respiratory sinus arrhythmia estimations in different applications. First, using the real respiratory recording, and also trying to remove this sensor and derive the respiration from the ECG. And I used this in different applications, but in the thesis I mentioned some of them. So in terms of aging, we found an increased respiratory sinus arrhythmia level in younger populations. This is not new, this is not a discovery, this has been already suggested in previous works, but the idea was to see if different methods could capture differently this change of the respiratory sinus arrhythmia. We also did an application in which we quantified the respiratory sinus arrhythmia using a wearable device. And we showed that the quantification of the respiratory sinus arrhythmia was better to capture changes during the day of the activity of the cardiorespiratory coupling compared to some standard methods. We also applied the respiratory sinus arrhythmia estimation and the ECG-derived respiration in people with heart issues. And yeah, the quantifications are very affected by irregular heartbeats. So it's important to, I think, study a little bit more this effect and to try to understand a better way to do the interpretation in these cases. And we also applied the methods to study a data set of people with sleep apnea problems and sleep apnea comorbidities. And we found out that the respiratory sinus arrhythmia increased in unhealthier subjects, which was an interesting finding during this PhD. So as a conclusions of this part, it looks like the respiratory sinus arrhythmia is overactivated in diseased populations. The EDR and the respiratory sinus arrhythmia quantifications are significantly affected by irregular heartbeats. The respiratory sinus arrhythmia is possible to, it is possible to calculate it using the respiration derived from the ECG, and also we can capture different information than the standard parameters. And yeah, that's it for this objective. So just to summarize, in terms of the quantitative evaluation of the respiratory sinus arrhythmia, we developed a method that is available online that can be used to compare any linear method for the quantification of the respiratory sinus arrhythmia in a quantitative way. We studied the methods in uncontrolled conditions and unhealthy populations, and we tried to see how the model order selection affected the respiratory sinus arrhythmia quantifications. We also have a method to study linear and possible nonlinear components of the respiratory sinus arrhythmia. And the methods were used to study different conditions, especially in ambulatory monitoring. As a future work, the model could be adapted to also include nonlinear interactions, and some more methods could be compared. For methods in uncontrolled conditions, I think more populations need to be studied, not only the ones that I did, but we also did some other works, but the samples were too small to try to conclude something or to say something. So I think it's worth to investigate more populations in the future. Regarding the linear or possible nonlinear components of the respiratory sinus arrhythmia, the method seems to work, but also we didn't find a significant nonlinear interaction. So I think that it is important to investigate more populations and more conditions, and also to better understand the tuning of the models and the mathematical tools that are used for this method. And in terms of the use of the methods for ambulatory monitoring, again, I think it's important to study more populations and to study more applications and different devices. And that's it. Thank you for your attention. And yeah, this is the moment for the questions.
So thank you for your presentation. We are now moving to the second part of this topic defense, which is the questioning part. Thank you. I will briefly introduce the members of the jury to you, and I will do it in the sequence we will take the floor. We are welcoming and give the floor first to Professor Luca Faes from the Department of Engineering from the University of Palermo in Italy. Then comes Professor Raquel Bayon from the Department of Electronic Engineering and Communications, University of Zaragoza in Spain. The other members of the jury are members of KU Leuven, different faculties and departments. First, Professor Kaat Alaerts from the Faculty of Movement and Rehabilitation Sciences. Then Professor Dries Tesselman from the Faculty of Medicine. Professor Chris van Hoof from the Department of Electrical Engineering and INEC. Professor Carolina Baron, supervisor from the Department of Electrical Engineering. With the supervisor also Professor Sabine van Uffel from the Department of Electrical Engineering. So, I first give the floor to Professor Luca Faes.
Sorry.
Thank you. Thank you for the introduction also. First of all, I want to congratulate with the candidate for the work done both in the thesis, which I appreciated a lot reading, and for the presentation of today. So in your work, you identify open questions that are very relevant for physiology and the clinics. And you address these questions proposing several novel methods and also comparing these methods with existing one for the evaluation of respiratory sinus arrhythmia. I have to say that I also liked in your presentation the analogy that you draw with coffee and rain.
Okay. Nice.
Not only because probably it's good for you coming from Colombia to do this analogy, but also because it is a nice way to show that data-driven approaches like the ones that you develop can be defined and used in an understandable way. Making a reference to that analogy, in one of your slides you show which are the various steps that lead to the production of coffee, starting from the coffee plant up to the cup of coffee in your table. If you want to translate this analogy in the study of respiratory sinus arrhythmia, can you, based on the results on your thesis, understand which are the mechanisms that lead to the generation of respiratory sinus arrhythmia? So, can you draw some conclusion about which are the physiological mechanisms that derive, that generate the phenomenon that you observed and quantified?
Mm-hmm. Well, based in my results and the analysis of the signals that I did, I'm not sure if I can conclude something specific about it. But from literature and from the studies that have been done before, we think that there is some mechanical effect, of course, but there are also some the connections from the central nervous system, as I explained, but also the baroreceptors, chemoreceptors in the aorta and in the heart and in the veins as well. And this is supported also from these studies in dogs and cats that I mentioned, in my thesis, in which the mechanical effect is isolated, is like removed, but the respiratory sinus arrhythmia remains even after removing this mechanical effect. So yeah, just to answer, I am not sure if I can support specifically one type of modulation of the cardiorespiratory coupling with the methods and my results, but from literature we have some evidence that the respiratory sinus arrhythmia occurs from a mechanical point of view and also from other modulators of the body.
Okay, and yes, another aspect that can be considered is that you do in your thesis an assumption that respiratory sinus arrhythmia is an unidirectional mechanism whereby respiration drives the heart rate. And again, this goes well in parallel with your analysis in which, in your example, sorry, in which you see that rain can drive the production of coffee, but not the opposite. But I was wondering whether this assumption can be really verified in the physiological systems. Due to the complexity of the human regulation, it could be also possible that you find a relation that goes along the other direction. So, did you investigate this first in your work, or
no.
can you make some reasoning about that?
Yeah, I didn't investigate this. It's a possibility, of course, but your methods, the information framework could be used to evaluate this if we have an interaction, like in the direction of the interaction. So I think we could use them to do this comparison, but also maybe, for example, with subspace projections, if we do the inverse, if we use the past of the heart rate variability to estimate the presence of the respiration, for example, maybe we could have also an estimation of the coupling in the inverse direction, maybe. It could be, it's worth investigating, maybe some methods like this one, but like apply it in an inverse way and try to see the direction of the interaction.
Okay, and another issue is there related to the comparison between linear and nonlinear methods. If I'm correct, you in your analysis, the one related to sleep apneas, if I'm correct, you identify the linear coupling as the dominant one. Is this the only context in which you compare the linear and nonlinear interactions? Maybe in different conditions you can find that this modulation is more nonlinear.
Not myself, but Carolina did a work of in a data set of people during autonomic blockade, and in that case there were some significant nonlinear interactions. So yeah, in this case, during different sleep stages, there was no significant. The nonlinear part wasn't significant, but that's why I say that maybe we should study more conditions and more diseases, and to see if in some other cases we can find some more.
And can these results be related to the method that you use? Of course,
it could be.
If you use different methods to detect non-linearity, you can get different results.
Yeah.
So
actually, one colleague of mine, Andrea, which is also here, she applied a different method in the same data set, and she found some significant non-linear interactions. So yeah, definitely, I think that more research needs to be done in the methods, and like in understanding how for example, the tuning of the different parameters and how they capture differently the interactions. So yeah, like in the same data set, she found non-linear interactions.
And how did you detect exactly the presence of non-linearity? Did you use surrogates or through a comparison between linear and non-linear?
No, it was, I did both, comparing the differences between the linear and the non-linear interactions, but also analysis of surrogates to see if the non-linear interactions were actually significant or not.
And in both cases, you found that the contribution was not so relevant.
The surrogates told me that the nonlinear part was not significant, but when you compare the linear and nonlinear estimator, in that case, sometimes they are significantly different. Mhm. But again, this could be also correlated with the tuning of the hyperparameters. So I think it's important
Yeah, I think
to
it's
investigate.
highly dependent on the methodology that you use. Okay, I think.
Okay, thank you, Luca.
Okay, first of all, I want to join also the congratulations to the PhD candidate.
Thank you, Raquel.
And I would like to emphasize from the work that you have done, the number of publications that you have that support the quality of your work, that you have used simulation studies, but also real data, and that in the real data you have worked with very different databases. Because facing a database usually is challenging and takes a lot of time to understand the signals, to do the pre-processing, the cleaning, the understanding. So it is something to emphasize that you have worked with so many different applications coming from clinical studies, from ambulatory recordings. So I just wanted to make this point.
Okay.
So I find your research very interesting, and in fact I have a lot of questions, and I will have to select just some.
Okay.
Okay. So I have enjoyed a lot the study of the influence of the model order. So if I understood correctly, your proposal was to use always the same model order. And then my question is, but given a specific application, how do I select the model order to use?
What I would do in that case, maybe you could. That's a good question. I have to think. I mean, according to my results, I say that you should fix it because if you fix it, you are able to detect different levels of coupling, and that's what you want at the end. If you want, for example, to see how the coupling changes between different sleep stages.
And
I think that maybe we can do different simulations of the different conditions to try to see how the coupling, the model order affects, like changes the quantification that we obtain in different conditions and give some recommendations. But the thing is that if you have the data set with the ECG signals and the respiratory signals, you are already computing the coupling from that data set, but you don't know if it's an effect of the model order or if it is the actual coupling value that you want to evaluate. So I think we need to find a way to also simulate conditions like apnea or some other conditions and give also recommendations of the best model order in a specific case. That's what I think at this moment.
Okay, because one possibility would be to obtain the optimal model order with different approaches, like classical approaches, or the one that you proposed, which is called the empirical approach, based on respiratory signal. And then you can say, I will use the minimum model order for all the subjects, or the maximum, or the median, or... Do you think this is something that makes sense, or it's?
You mean like computing every time the best model order? Well, actually we did it, but the problem is that when you use different criteria, like the minimum descriptor length or AKIK or the one that we proposed, the model orders that you obtain are not consistent one to each other. Like one can give a very high model order, and some other can give a very low model order. So I think it's not, with this inconsistency, it's difficult to really choose only one. That's why I think fixing the model order is the best alternative, and then you can distinguish actually how the coupling is changing. Mhm.
Okay. Then another thing that was related to the performance of respiratory sinus arrhythmia methods was the effect of the change of phase between respiration and heart rate variability. And you give very clear recommendation of up to what level of phase change is allowed in order to obtain reliable estimates. But how would you again handle this? Would you try to measure the change between the signals and then disregard those measurements whenever the phase of the signal is higher to this limit? Or how would you take this to a practical application of the method?
I would, if I detect a lot of phase changes, then I wouldn't use these two approaches because they are very linear. So I would go for some time frequency representation, and because in that case I will be able to see the coupling no matter the phase change.
Yeah.
I think it was worth to investigate these methods because computationally they are not that demanding, and this is important also for wearable devices and mobile monitoring. Time frequency analysis require more computational power. So yeah, like that would be the next step, I would say, but they will require more computation. So.
And this was related to one of the other comments that I have, and is that in these cases in which, for example, the model order selected by different methods is so different, or when you have phase changes, do you think that it is still worthy to apply this method, or maybe it's better to go to these other methods to evaluate respiratory sinus arrhythmia?
I think again it's dependent on the application. In this case, for example, I studied the methods mostly in stationary segments, and but then if you have non-stationarities, maybe these two methods again will have problems to capture the respiratory sinus arrhythmia correctly. So then in those cases you should go for some other approaches, or you should find a way to adapt these methods like subspace projections but in a moving window, or but that we tried as well a little bit during my PhD, but again the selection of the model order was super challenging. So yeah, I would say it depends a lot on the application.
And I had another question that I think it's a general question regarding the use of respiratory sinus arrhythmia. You have made a study of different methods, proposals, where to use one method, where to use the other, what to take into account, and you made a very comprehensive study. But I wonder if these measurements of respiratory sinus arrhythmia are enough, or would you also need to take into account specific characteristics of respiration? For example, the respiratory rate, the amplitude of respiration, the waveform, in order to obtain a more accurate estimate of the functioning of the autonomic nervous system. This is something that is not done. I haven't seen done usually. People are usually estimating respiratory sinus arrhythmia as it is. But so this is just to know your opinion.
In the case of subspace projections and cross entropy, I think there shouldn't be, well, at least in the case of subspace projections, let's say like the amplitude, for example, of the respiration shouldn't be that important, because at the end you have a ratio of the power of the respiratory component and the power of the heart rate variability itself. So in this case, at least in terms of the amplitude of the respiration, you shouldn't have an effect. With cross entropy, maybe it's the same case, because at the end it's a regression, and if the amplitude of the respiration changes, I would say the coefficients of the regression shouldn't affect the errors at the end to estimate the cross entropy. For the other methods, I would have to think, but again, I think it depends on each one of the methods. Like, we would have to see each one of them and see how the characteristics of the respiratory signal affect the computation of the parameters. Also, this is a set of parameters that I used, that I compared, but you can actually extract more parameters using the same approaches. So maybe in some cases there is some more parameter that you can extract, and that would be more or less sensitive to the respiration. So definitely something to study, but in each specific case, I think you will need some different reasoning.
Okay, thank you, and congratulations again.
Thank you,
for
Raquel.
your comments.
Thank you. So, hi, John. First,
Hello,
I
Kat.
would like to follow my colleagues in congratulating you
Thank you.
with what I think is really an impressive line of work. Also, very nice presentation. Thank you for that. Although I've not been really connected to your PhD from the start on, we got to know each other over the past year during some monthly meetings that we had, and I became a bit more acquainted with your work on RSA. And I always thought that the meetings were very interesting and very inspiring, so thank you for that. Actually, with my following questions, which will be a little bit more out of domain, I would like to engage with you a bit more in a discussion towards potential translational perspectives that you see connected to your work. And some of it that you've, of course, already covered. So maybe a first question. So in chapter 6, you applied your methods also in patient populations, most of which were patients with severe obstructive sleep apnea. And here, strikingly, you actually showed that quite consistently that RSA was actually higher in the most affected patients. Mm-hmm. To me, this would actually be quite counterintuitive, because mostly the RSA index is really associated with better cardiovascular health. So could you maybe comment a bit on some interpretations that you see connected?
Yeah, there are different hypotheses of the function of the cardiorespiratory coupling. One of them is that it is a mechanism to optimize the use of energy. We hypothesized that maybe this mechanism is overactivated in these cases in people with a high apnea and with cardiac comorbidities to compensate for the extra energy that they need to use. But again, this is an hypothesis based on these biomedical signals, non-invasive signals. It's difficult to say that this is actually the case, but I mean, you can see, despite that the differences are not significant, which is also important to mention, but you can see how it looks like it is increased in unhealthier populations, and this is the hypothesis that we proposed. But to be sure about it is not that easy. You need some more studies. Maybe you need to go more deeper, like with some other signals or with some experiments in animals or.
Yeah, actually, maybe perhaps a bit of a related question. It was a notion that you introduced in your introduction, where you indicated that there is this distinction between involuntary, more unconscious breathing and voluntary or more conscious breathing. And that you also indicate that, yeah, especially the involuntary breathing is associated with more vagal innervation and control centers of the brainstem. And I was wondering, to which extent do you have any data where you ascertained the type of breathing that was recorded, or are you aware of any studies that specifically looked at the effect of either voluntary versus involuntary breathing patterns on RSA?
Yes, there are some studies, and I think I read a couple of papers in which they control the respiration at the beginning of the recordings, and then they let the people just breathe as they want. But I don't remember exactly the results. But we can see that the voluntary control can override the rest of the activity, and you can see it, for example, in meditation when you can see very clearly how the respiratory rate moves a lot to the low frequency band of the heart rate variability, maybe. But to which extent, I'm not totally sure.
Yeah, because if it's indeed only the involuntary part of breathing or control of breathing that would be activated by the vagal control centers, then you would anticipate RSA to be higher during this type of conditions, right?
Yes, that could be, but also the dependence on the respiratory frequency, because if you breathe slowly, then the coupling is also going to be, you will see it in a stronger manner. So I would say it also depends on the respiratory frequency, for example.
Okay. Then maybe another short question in between, because I saw that next to your main doctoral work that you also contributed to some other side projects
yes.
that you included in your appendices. And one was actually to analyze, to develop new algorithms to analyze pressure signals recorded from a stomach balloon.
Yes.
And then here with the main goal to identify a motility index. But I saw there, and I thought that was interesting, that also with this pressure device, you seem to be able to capture some of the other frequencies of the heart and respiration.
Maybe.
And I was actually wondering, do you think that these type of measurements could maybe contribute to characterize different aspects of cardiorespiratory coupling? For example, the mechanical component that you indicated.
Yes, I think so, and also because you put the balloon somewhere here, then of course the pressure components of the lungs and the heart also affect the balloon there. So something that I proposed at that moment was to use these signals to also try to see the cardiac and the respiratory activity. They are inside, so maybe the effect of the let's say delay of the electrical activity and the recording in the ECG maybe would be less present. So yeah, I thought a little bit about it, but I don't know, maybe in the future we can explore it a bit more. How to use that device to also evaluate cardiorespiratory coupling would be very interesting.
Did you also perform ECG recordings of these
No,
participants?
no. Respiratory recordings, yes, but not ECG recordings.
Maybe in the next side
maybe
project.
That would be that would be very interesting, I think.
Okay, maybe then, so my final question, a more general one. Probably you and many people of the audience, we know that we are all bombarded to some extent with different applications and smartwatches that can record any of our physiological signals of the body, including also heart rate and HRV. and that these are then for assessing states of stress or relaxation or even sleep quality. And I was actually wondering, considering your expertise in this domain, what is actually your opinion about the quality of these devices, and how do you think they should evolve in the future to be able to more applicable for what they pretend to measure?
I believe they should be useful, and especially in the future. In Colombia, we have very isolated regions, and we will explain later the joke. But the thing is that hospitals cannot reach sometimes these regions, and using this type of technologies that could be useful for this problem, for example. But also in Europe, people are getting older, like the population, the pyramid of the population shows that each time there is a less proportion of young people, so you also need to move the hospital out of the hospital and try to provide healthcare at home. In this regard, I think these devices are useful. Some of them, of course, are more affected by noise than other ones. PPG, as you know, sometimes is very affected by noise, but also this morning with Raquel, and Raquel showed some examples of how PPG can be used to extract the respiration and to analyze the pulse rate variability. So I think they have a lot of potential. We still have a lot of challenges in terms of noise, as you say. But yeah, I think that's my opinion, maybe. I see a lot of potential. That's why I keep working on this.
Yeah, then maybe as a follow-up, maybe last question, if you consider all your line of work, what do you think was the most important thing that you worked on, or perhaps formulated differently? If you would live in an ideal world and you would have ample time to continue to work on a particular part, what would be that part, or how do you see then future steps in that?
Well, my main project was in the study of the cardiorespiratory coupling, and I would try to go more for the applications. The thing is that, a PhD doesn't go smooth. So I started with some applications. I got a bit frustrated because you don't find good things. Then I said, also because we had a conversation once with Raquel, and then I came up with the idea of the simulation, and then I found out that the simulation was a very good idea to help to better understand the estimations. I would try to first do the model, like in the first year of my PhD, and then the rest for applications to try to understand what I see in the simulations, how it is actually reflected in all the applications. Here we have many challenges. We don't have a lot of data. The recordings are noisy sometimes. I had some other data sets, but there was one of people during dialysis. The noise was super difficult to handle, so we couldn't find something interesting, let's say, and the sample was a bit small, so it was difficult to conclude something. In your study as well, it was difficult during autism to find some significant, or we didn't find a lot of significant effects in terms of cardiorespiratory coupling. So, I would try to explore more the applications.
Okay, thank you very much.
Okay. Thank you.
Okay, John, I also want to congratulate you with your excellent presentation and the huge amount of work that you performed for your thesis.
Thank you, Liz.
I think it's a very difficult topic that you worked on, but it's also a highly interesting topic, so very, very nicely done.
Thank you, Liz.
I have a few questions also for you. Maybe I would like to come back on a question of the previous jury member with regard to the obstructive sleep apnea. As you know, I work in the sleep lab, so it's a little bit related to my daily work. I would like to ask you how you see the use of the RSA signal in the clinic in the future, because you mentioned that the RSA maybe changes in patients with sleep apnea, but how can we use it clinically? To diagnose these patients or to do other evaluations in these patients?
Both to maybe diagnose some diseases, to also detect the degree in which a disease is affecting a person as well. But I also think that we still have to understand more how the respiratory sinus arrhythmia changes in different populations. In many studies, for example, when you study people with cardiac problems, sometimes it's not very clear what they do with the irregular heartbeats. And something that I show here in my thesis is that even after doing correction of irregular heartbeats in the ECG signals, you have some significant deterioration of the quantifications. At the same time, the irregular heartbeats, we assume that they come from innervations, like extra innervations of electrical innervations in the heart, but maybe they also come from the central nervous system itself. So yeah, like we first need to understand how to interpret the estimations in different populations, or as we did in the study of sleep apnea, to only use segments of the signals in which we don't have noise, we don't have irregular heartbeats, and we don't have all other confounders. And in this case, for example, using always the same sleep stage, like trying to eliminate all the possible confounders that we could have.
Okay, I agree on that, but don't you think that also in the irregular heartbeats and in the epochs with breathing stops also lies a lot of information that we can use?
Also what?
Also lies a lot of information
yeah,
that we could use.
Of course, but it's more that from what I showed, having, like, if you have irregular heartbeats, for me is more that you don't know what is actually happening with the cardiorespiratory coupling. But if you have segments in which you didn't see irregular heartbeats or noise or whatever, then you know at least that this quantification is more trustworthy than in the segments with irregular heartbeats. So it's like you are evaluating actually how the cardiorespiratory coupling is, more than the effect of the irregular heartbeats.
Okay. I have another question with regard to the use in the clinic maybe, but do you have an idea on the variability or the stability of the RSA signal in a patient if you measure it today in a certain state of the patient when he is sleeping in stage 2, and you measure it within a week again? Do you think that we will have the same number, or do I see it wrong?
I didn't study that, but I think it's a very interesting thing to try to explore. Maybe with the data of the SWIT study we could try to have a look, but the problem is that we don't have the sleep stages, so we would have the...
Yeah, or during the same activity during the day, I don't know, you can think of many situations. But I'm just meaning, is it the same number for a given patient in the same situation, or do you think that it has a lot of variability?
No, I think it's something to study. Like from a physiological point of view, I don't dare to say something. It's something to study, I think, and to try to see Also, you have the seasons, for example. You have changes in now. It gets dark very early. So, like 2 weeks ago, it was different. So, I think in that case, our respiratory sinus arrhythmia will be different now than 2 or 3 weeks ago. If you are in Colombia, we don't have seasons, so maybe it's going to be more stable throughout the year. Well, for women, you also have the cycle. So the hormones are different. So also depending maybe on the day of the month in which you evaluate the respiratory sinus arrhythmia, you will see some change as well. There are many factors. So.
Okay, thank you. I don't have any more questions.
Thank you. Thank you, John.
Thank you, Chris.
Fantastic work. Great to see the number of publications, the output. Fantastic presentation. And with one exception, you talked about coffee, and all we had was some water.
That's true. I should have.
Yeah. Especially Colombian coffee. Regarding the work you did, analysis of some of the SWEET study data, you mentioned that a split was made in 5-minute epochs. Now, we struggle, and I'll come back to that, we struggle with experiments in free living conditions because of all the artifacts and so on. Do you think there would be a better way or a solution to that? Do you need shorter epochs? Or how could you make more sense of the data in the presence of all those confounding factors?
Well, in terms of noise, for example, we first need, I think, to eliminate noisy segments. There is no other alternative. In particular, in the SWEET study, the quality of the signals was very important because we did the derivation of the respiration from the ECG, and in those cases, you need to guarantee that the signal has enough quality. I think it should be possible to do the cardiorespiratory analysis. In the thesis, I also show that cardiorespiratory coupling captures better the change of the coupling during the day than the more standard parameters. So there is definitely potential there. But at the same time, the idea of the SWEET study was to evaluate the stress. This is not in the thesis, but I didn't find significant differences between different emotions and stress levels in the dataset. And this is, of course, due to the fact that it's a completely uncontrolled experiment. And also that there were very few samples of stress moments. I always say this, but I think people in Belgium and in the Netherlands are very happy at work because there were no periods of stress in the signals. But then at the same time, this makes it difficult to try to conclude something from the study. I think that we need to better understand how the signals change, but for this, I would go for a semi-controlled experiment in which we have both some periods of some controlled experiments and some periods of non-controlled recordings to try to see how the signals can give us information in different moments.
Yeah. Mm-hmm. Yeah, you, with your last two sentences, you answered my second question without me asking it. And because you observed, on one hand, you said it's possible that ECG signals are more contaminated. On the other hand, there can be indeed other interactions. And like whether it's talking or other factors. I was wondering, I agree with you that semi-controlled would be a far better route to go than completely studies in the wild, because there's so many unknowns, and then you have to hypothesize or in practice remove data. And I think it's really, that, for that reason, it's the right approach. Would it make sense to do studies where exactly you look at just those confounders? Like, what is the effect of talking or sneezing or other things, so that you can also model it?
Yes, like yeah, I think that's a good alternative to see. But
it doesn't sound very exciting. But on the other hand, if that helps in the analysis,
yeah. But the problem that I see is more to isolate specifically those confounders. You would need to do the experiment the same time of the day, maybe the same day of the year, maybe with people in the same range of age, the same gender, you know. So then it becomes, you will have a very small sample at the end, or you have to do this experiment in Colombia at least, because then you will have the same weather the whole year. And then you eliminate one in that case.
I'm very happy you contradict me, because I also don't believe in it. I don't think there's a standard sneeze either. It's towards variability. Thank you.
Thank you. Congratulations, John. I will congratulate you more later. So I will go directly to my questions. So sometime, well, even at the beginning of your PhD, you started working on the cardiorespiratory phase synchronization. And I remember you were a bit frustrated with it. So,
a bit what?
Frustrated? Frustrated?
Ah, yes, I was.
So I wonder if we should, like, if you advise us to stop looking into that direction, or under which conditions can we continue looking into that?
The problem with the cardiorespiratory phase synchronization is that you need to know this instantaneous phase. And finding that instantaneous phase is not trivial. You can use the Hilbert transform, but for the Hilbert transform, you need narrow signals with a narrow band. Otherwise, you cannot trust what you get. You can also detect, for example, the respiratory onsets, like the peaks, and then assume that the phase, an instantaneous phase in between. But again, in some cases, for example, when you are talking, that instantaneous phase is also not trustworthy. I think it's worth investigating, but in conditions in which the respiration is more stable, like during sleep. But again, during healthy sleep. Because if you have apneas, then it becomes again a little bit difficult. In my case, I tried to study the cardiorespiratory phase synchronization in the Fantasia datasets, and it was very frustrating, as you say.
Okay, and then related to this phase changes, so we've seen recently some papers about the negative RSA, which relates a bit with your study of the phase differences. So how can we approach that? How can we start analyzing that if there is something in there related to this negative effect that they are reporting?
So in these cases, I think the analysis needs to be continuously. So we would have to go for either a time frequency representation or adapt, for example, subspace projections to be able to do the continuous evaluation, which is something that we tried, but it didn't work very well. And then try to see or to correlate these phase changes with, I think from what I have read, it seems like this happens in more critical conditions, like in more severe conditions.
Like patients.
Like, for example, with the signal of Pablo in the intensive care unit, that we saw it, that Pablo was obtaining a very low quantification of the coupling, but it was because of these phase changes. So it would be worth, I think, checking in some continuous manner and see how it changes in these specific critical conditions.
Yeah. Okay, thank you very much.
Okay.
Okay, John, I will also postpone my congratulations to you and start with some difficult questions because I'm the last one.
Okay.
Okay, I would like to go also some broader in the field. And my first question is related to what we heard this morning. So you're studying cardiorespiratory coupling, and I really like it. And you show that these signals are coupled, and especially the RSA phenomenon. But this is not in isolation. So there are many other physiological signals which also contribute, in fact, to the coupling. Can you study, why did you, or what other information can you get by going more towards network physiology instead of studying RSA in isolation and only looking at the cardiorespiratory coupling? How can you generalize your algorithms, in fact, more towards network physiology? And which other signal would you take as the next one to be involved in this coupling?
Yeah, like in terms of the methods, for example, well, I used only cross entropy, but Luca already showed this morning that the whole information framework has a lot of different measures of the coupling, so we could use that as well. With subspace projections, I applied the method for the cardiorespiratory coupling, but in principle you can use pasts of different signals to try to predict present samples of other signals. So in that case you could also study coupling between other systems. And about the signals, well, it would be interesting to include, for example, maybe the blood pressure as well. Raquel also mentioned this morning that actually the PPG or the pulse rate variability can reflect some other things, so it would be worth including it. With Kat and Javi. With Kat and Javi is studying also brain, heart, like different coupling between different oscillations, and different waves in the brain could also be correlated with some other oscillations. So maybe EEG and in some alpha, beta, or some other bands could be interesting to study. So.
Okay, thank you very much. I have a last question. Is in fact how to extrapolate your methods to neonates. You're working in neonatal. Well, in the NICU, with neonates that have a heart rate, of course, of 120 beats per minute up to 180. Also, it's a big problem. There is also this coupling to respiration, RSA. That, of course, you are in a totally different range, in fact, of the heart rate. So, which of your methods can really be extended to that range?
The main problem here for me is that babies, well, first of all, the signals are not easy to record from the babies, but also sometimes the respiratory rate seems to be too fast. And the ECG signal is like a sampler of the respiration, no? And in most of the cases, the heart rate does not go as fast as you would need to be able to sample the respiration in a good way. And I don't know if you remember, but with Mario we tried a little bit some signals of babies, and there was no respiratory sinus arrhythmia, apparently, at least with these methods. But saying that the respiratory sinus arrhythmia doesn't occur in babies, I don't know if it's a good, I mean, it's something that I cannot answer that easily.
Surely it occurs because I discovered it already 20 years ago
Okay.
in some of these signals.
But at least with these methods, like since you trust on these two signals, cardiac and respiratory signals, the way, like the speeds of the respiration in the babies makes it very difficult to use the methods to analyze the cardiorespiratory coupling. So maybe you need some other signals, more the EEG or.
Okay, thank you very much.
Okay.
So this is a public defense, and I have to give the opportunity to anyone in the audience who is willing to ask an urgent question. This one? Is this publicity or? So if there is no question, the jury will convene in closed session, and you will have to be patient for a while.
There should be there.
Okay. And then when we come back, you stay here, and we will stay here. And we read something, we graduate, and we sign the fact that I give you a professional certificate. This is the final one. I found this one standing there, so I don't know if it's
We can ask. I can ask.
it was here.
Okay.
Very good. Really like it. Kudos for the coffee example. That was really the best. That was really the best. Let's warm it up the voice.
Congratulations.
Thank you, please. Thank you.
Can I have a...
Of course,
Yeah,
take it.
Okay. Oh, is it the last one? Or?
Yeah, it is the last one. That's okay.
Okay.
And thanks for being here.
Yeah. Well, thanks for the invitation. A colleague was also joining online. So,
Okay.
Yeah. So he also, I think, followed before noon. But yeah, I couldn't come.
Okay, we recorded the symposium, so I will make sure that you can have some access afterwards. So you can see it.
That would be nice.
Yeah.
Yes.
Okay,
Cool.
thanks for being here.
Yeah,
Dames en heren, geachte collega's, ik geef u kennis van de uitslag van de beraadslaging van de examencommissie aangesteld voor de proef van dokter in de ingenieurswetenschappen. De heer John Freddy Morales Tellez heeft aan de faculteit een proefschrift voorgelegd handelend over Respiratory Sinus Arrhythmia Estimation, Closing the Gap Between Research and Applications. In het Nederlands luidt dat als schatting van de respiratoire sinusarrhythmie, de kloof tussen onderzoek en toepassingen dichten. En dit tot het verkrijgen van de graad van dokter in de ingenieurswetenschappen. Hij heeft het proefschrift in een openbare zitting voor de examencommissie verdedigd. De examencommissie heeft vastgesteld dat zowel aan de wettelijke en decretale bepalingen als aan de voorschriften van het universiteitsreglement ter zake werd voldaan. En na beraadslaging verklaart de examencommissie dat de heer John Freddy Morales Tellez voldaan heeft aan alle eisen voor de proef en de graad van dokter in de ingenieurswetenschappen behaalt. Mr. John Freddy Morales Tellez, the examination board has concluded that you have fulfilled all the requirements laid down in the university regulations for the doctoral examination. And therefore, on behalf of the rector of KU Leuven, I confer upon you the degree of Doctor of Engineering Science, which is PhD, in Electrical Engineering. Bijgevolg. En in naam van de rector van de KU Leuven verleen ik u de graad van dokter in de ingenieurswetenschappen richting elektrotechniek. Daarmee is de zitting gesloten. This closes the session. Be seated, please. I give the floor to the supervisor. Supervisor.
Okay, dear John, congratulations, Dr. John. Do you realize you reached your end goal? That's the PhD degree, and you know that's the highest degree you can get at university. So it's a great pleasure and honor for me, and as promoter and Carolina as co-promoter, and of course the other members of the committee, to congratulate you and to see that you reached this stage after well, I would say almost 4 years and a half of research work, hard research work. This occasion, let me first thank Carolina, the co-promoter, for all the efforts she did to guide you on this research journey up to the end. Truly said, she is really the main supervisor. She made my life much easier, because she did the first line in-depth revisions of all your papers. So also I would like to thank Raquel and Luca, who did all efforts to travel to Leuven in these not so easy circumstances, I would say, but also for sharing their ideas with you at the benefit of your PhD research. I leave it to Carolina to elaborate more on this in her letter of thanks. In addition, I want to thank Chris van Hoof, and the IMEC teams you collaborated with. After all, it helped you to work out your ideas on cardiorespiratory coupling in wearable sensing applications such as stress at work and sleep. For the latter field, for sleep, I want to thank Dries Testelmans and the Sleep Lab of UZ Leuven for sharing their sleep recordings of OSA patients, which helped you to validate your cardiorespiratory models towards clinical applications in sleep monitoring. Where is Dries? Ah, yeah, Dries. And I also want to thank Kaat Allaerts, Kaat and her team for setting up a nice collaboration with our group. And finally, I would like to thank Herman Neukermans for his willingness to share this PhD defense. So, John, you started your PhD in July 2017, I would say, 2017. But in fact, I selected you already much earlier as a potential PhD candidate, based on your motivation, your sweet character, and excellent results obtained during my master course on biomedical data processing, for which you got a 19. So, moreover, moreover, because of my nice experience with Colombian students, PhD students with Carolina and Alexander Caicedo as nicest examples, I became biased in my judgment and expected Colombians to perform in an excellent way and also create a very nice atmosphere in the group. Moreover, I learned to know you better during our preparation for the industrial internship in the gastrointestinal lab at Johns Hopkins University in collaboration with trans-stimulation research. In addition, you succeeded to select one of our master topics under my supervision. So I had plenty of opportunities to get to know you and know that you were, in fact, one of my favorite PhD candidates as soon as an adequate budget would become available. And this was indeed the case. So we got a C3 project with Target, which was approved, as well as a B project with IMEC on smart health monitoring using wearable sensors. Nevertheless, we asked you to prepare an FWO SBO grant application on cardiorespiratory coupling for stress detection. And I remember how stressed you were during the trial sessions we set up. Working on stress generates stress, new stress. You still had to learn a lot on presentation skills and science communication at that time. Luckily for us, you remained motivated when you didn't get the grant. So in these 4.5 years, you showed and also realized your ambitions you wanted to achieve in your life. Originally, you aimed an academic career, and therefore, you did a lot of efforts to improve your presentation skills. I was not so convinced the first year, and my reactions made you very sad. I'm really sorry about that. But almost 2 years ago, you followed the course Discovering Your Career Profile. And you asked me to describe your 4 strengths and weaknesses in a professional context. So I'm very happy to point out as your strength, still valid today, perseverance, soft-hearted, super friendly, never angry, and trustworthy. Regarding your weaknesses, I prefer not to repeat this here. In fact, if I have to redo the exercise now, I'm happy to tell you that you succeeded to remove your weaknesses. You clearly showed leadership as responsible teaching assistant of the most difficult session of my course, covering chapters 4, 5, 6, and 7 of my 2 courses, biomedical data processing and human health data processing. And you were a very dedicated and excellent daily supervisor of numerous master theses and biomedical design projects. And mostly you had to guide the most problematic students. But you always did it with great esteem for any progress the student made. Thank you so much. We saw you really growing the last 2 years to a mature researcher, thanks to your perseverance and great efforts you did to realize your dreams. However, now that I'm convinced that you could indeed start an academic career, you changed your end goal and decided to move more to industry. In fact, not really, since you decided to go to IMEC, provided you will be accepted there. I'm not sure. You have to tell me. Furthermore, I would like to thank you for the nice atmosphere that you brought into our group, Biomed. Truly said, I think you are one of the beating hearts and best team players in Biomed. And I'm afraid we may miss that so much after your leave. As far as I remember, you were always enthusiastic, excited, and I always saw you with a happy face, enjoying all Biomed and ESAT activities we organized, including the sports days and the Christmas parties. I appreciated very much your last interview in the journal Geniaal of our faculty, where you described your experiences as an international PhD student in our faculty. To my surprise, I read that you consider independence and learning opportunities, and especially the non-technical ones, as the most valuable possibilities offered to you in ESAT. In fact, you have grasped all opportunities we offer to you. We ask you to start to develop the motility index for one of the spin-off companies of Target, Vipun. You combined this work with an intense collaboration with IMEC in 2 projects, a B project and a so-called AAA project, on the topic of wearable cardiorespiratory health monitoring, together with Willemijn Groenendaal as key coordinator. Then you also accepted to guide Hélène de Cagnière during her PhD towards the development of a cardiopulmonary fitness index for personalized heart failure management, and you cooperated with her, with Hasselt University and with ZOL. So I'm happy to read that you never felt discriminated during your KU Leuven period. Within Biomed, we try to create an open and social atmosphere. Diversity is one of the strengths in a research team. When we succeed to share our ideas, respect each other, and give everybody the freedom to be himself. However, I feel sad to hear that KU Leuven fails to help international students with mental health problems, as we could discover with the questionnaires or with the survey, in fact, that was made recently during the Corona period. Our dean, together with the diversity team, is aware of these problems and try to generate new structures and a better social network between the international and the Flemish communities to alleviate these problems. Testimonies like yours are very welcome. To end my comments on your interview, I must say that I very much like the advice you give to other PhD students. And I just say here what you said. Trust what you do. No one knows more about your topic than you, not even your supervisors. Well, I think you proved this during the questioning in your defenses. So Biomed without John will really make a big difference. In particular, I keep nice memories of the conferences that we attended together. While you were awarded the 2nd place in the Best Paper Award of 2019-2020 in the competition of the IEEE EMBS Benelux chapter. But even more, I enjoyed the multiple marriages of Biomed friends we could celebrate together, including the marriage of Carolina, Margot, Christos, and Lieven. So these were always nice opportunities to have fun and dance together. Next month, you will leave ESAT, which was your home for almost 4.5 years. And you will take the next important step in your academic career, namely working in IMEC, I guess in a new country, although just a neighboring one, a new research environment, a new group. Well, John, I wish you a brilliant future in your career, but also in family life with Gibran. You have great potential to break through. Thank you very much.
This journey started during your master thesis on the detection of sleep apnea using the SpO2. This thesis was supervised by Margot. By Margot, and I remember that I asked her if I could join the meetings because on the one hand, it was on sleep research, and on the other hand, you are Colombian. So I was very proud to have a master's student from my beautiful country. The work you did during your thesis resulted in a poster that Margot and I presented in a conference. But it also gave us the opportunity of working with you and getting to know you, your hard work and dedication. I was convinced that you had all the right attitudes needed to pursue a PhD. So when Sabine asked me if we should hire you, I did not hesitate. And we soon welcomed you in Biomed. Although I soon regretted that when you said in front of the whole group that you are from the city and I am from the jungle. Anyway, you started working on the motility index together with Nick Hulen and Peter Jansen. This was a very interesting collaboration because you are so calm, you speak very slowly and always relaxed, while Peter has a million ideas at the same time, and he expresses all of them with a firm voice at once. There was a period where you were always on the phone talking with him, and you were always giving the best of you for the culmination of the project. As a result, you developed the index, which gave us publications and even a patent. Thank you for that. During your PhD, you also collaborated with 3 amazing researchers, Raquel, Willemijn, and Kat. You managed to deal with many projects at the same time, and you even joined our ECG group for a little bit. Especially for the dinners and the educational visit to the hospital. Anyway, you were always professional, kind, patient, disciplined, and always a pleasure to talk to. Your work was so structured, and there was always a clear line, well, most of the times. You tried so many things, and many of them did not work out. And I could recognize you when you were frustrated because you came to my voice with the following voice. No, Carolina, no sé. Then I knew it was time to step in and change the perspective or work on something else. Our relationship was not only professional. We also became friends. We shared many special occasions, and it was always a pleasure to travel with you and experience different cultures, drinks, and landscapes. You also shared some time with my husband, and we will never forget your kidney stones. You will explain that later. Apart from the respiratory sinus arrhythmia, we also worked on various things together, like the dress we made for Jonathan. I remember the face of Sabine when she entered my office and saw me wrapped in a blanket and you were laying on the floor. She literally started running away saying, sorry, sorry, sorry. And I could not explain to her what was happening. I tried to say, it's not what you think, Sabine. But we couldn't stop laughing. We traveled to so many places together, Singapore, Italy, France, the Netherlands, Greece, Germany. And I think I speak for all of those who have had the pleasure of traveling with you. You are a great companion. We even made a piece of art together in Berlin for Alexander Bertrand, which I think you should appreciate more. John, you're often surrounded by good people, good wine, nice friends, and most importantly, you make your friends feel at home. Thanks for creating those opportunities when people can come to you and talk about anything. Unfortunately, not everything has been fun. You have gone through so many difficult moments during the past years, and as a real warrior, you have overcome every obstacle that has come your way. None of those have stopped you from caring about people and from being so dedicated to your PhD and your friends. John, ha sido de verdad un placer enorme trabajar contigo, conocer tu vida, tus amigos, tu casa. Eres una persona muy especial, un trabajador como ninguno, y es que todo el mundo te quiere. Tus padres deben estar muy orgullosos de ti, y es un placer tenerlos acá con nosotros. No es fácil estar lejos de casa, lejos de la gente que puede animarlo a uno, cuando la vida se pone gris. Tú has estado aquí para mí. Siempre me agradeces. Siempre me agradeces por muchas cosas,
pero ahora es mi turno de agradecerte por tanto que has hecho por mí. Quiero darte las gracias por ser ese pedacito de Colombia acá, por recordarme que los sueños se cumplen y que todos los colombianos que estamos aquí somos unos guerreros. Siempre me hace sentir como una campeona y me hace sentir muy orgullosa de lo que hemos conseguido. Muchas gracias de verdad. Now, our professional lives split for a bit, and it is time to wish you all the best in your future endeavors with Gibran, IMEC, and the Dutch. I'm sure they will love you as much as everybody from every nationality does. You will do an amazing job wherever you are, and please do not forget our times together and the beautiful memories we made. Thank you for everything, Dr. Morales.
I need some minutes. This is overwhelming for me. Okay, sorry. Having all of you here just confirms me that I have a second family in Belgium. Since I arrived here, I have just felt a love around me. Having your company during these 6 years has been very nice for me. I have to thank first Sabine for the opportunity of doing the PhD. Carolina, you know that I don't find all the words to just thank you for everything you have done during these years for me. From a professional and a personal point of view, you have been there a lot. So thank you very much for that. To the jury members, Dries, Kat, Raquel, Luca, it has been a pleasure to learn from you. You have shared a lot of ideas with me and your knowledge. You are my professors. You taught me during these years, and I really appreciate it. To Biomed, it's a pity that COVID happened. But guys, try to keep the good atmosphere. I don't know most of the new people now, but it's a very nice group, and you will have some very nice years here. Now, my Belgian family. Okay, since the master, since I arrived here, I have had someone nice, someone to talk to share good memories, to have a dinner together, to talk when I was having a problem, when I was sick. As Carolina said, I had some health issues during the PhD. I had kidney stones, I had a couple of surgeries, and many of you were there just going to the hospital or visiting me at home. And when you are far away from home, it can be very difficult. But I can say that it wasn't that difficult for me, thanks to all the nice people that were around me. I hope I don't forget someone, but I will start mentioning some of you. Bruno, you were my first Belgian friend. I have always counted on you. I'm very glad to have you in my life, and from the very first moment you were there. Roxy, it's a pleasure to have you here as well and to have you with Bruno. You guys look amazing. Marco, Vale, you know that from the very beginning you have been there, so I'm very happy for that. And Marco, you know, you guys are always there. Peris, you are the most chill person in the world, but also very caring, and I really appreciate that. Jorge, I also don't know what to say. You are amazing. You are one of the best persons that I know. Apeksha, Antoine, you are just so caring with me all the time, with everything I do, with opening the doors of your house all the time for me and being there for me. It's just amazing to have you. Esteban, you are also one of the chillest persons that I know. Yeah, the way you finished your PhD was just amazing. Grantita, from the beginning, it was also very nice to share with you. Sandra, which is also there. You were my family for 2 or 3 years in the house, in the Latin house, as we call it. Che, from the distance, some people in Colombia that are watching, I hope now. Also, thank you very much. Jonathan, also thank you for the coffees, for being there, for your admiration all the time. And Marion, Amalia, you guys, without you around, I don't know, I wouldn't have managed to finish this. So many times I was stuck and you were there with a positive word and taking me out of my negativity when I was frustrated. So thank you very much. Carolina, again, from a personal point of view, I don't know how to thank you for everything you have done for me. Like, you say that you want to do something for Colombian people, and you started changing the life of a little Colombian guy that can be grumpy sometimes, as you saw it, but you were always there and always advising and helping me in many ways. Andrea, I hope we can share more in the future, but you started very well. Congratulations. Alright. And finally, I will switch to Spanish. Pama, tenerlos ustedes aquí. Completa todo. Para mí es una gran alegría. Gracias por todos los esfuerzos que han hecho en cada etapa, por ser un ejemplo de vida y por siempre estar ahí, por apoyarme, por comprenderme y porque a pesar de que hay cosas que no entienden o que tal vez no comprenden de mí, pues siempre han estado ahí para apoyarme. Muchas gracias por ser ese ejemplo, por su humildad y por todo, de verdad. Gibran, estos días has estado aquí como nadie más, me has estado apoyando siempre. Yo he estado súper irritable en la casa, pero no te has rendido y siempre has estado ahí, y de verdad que esto solo me reafirma que estamos el uno para el otro, y solo me hace sentir mucha emoción de pensar en los años que vienen y en lo que vamos a seguir compartiendo. Muchas gracias por estar ahí. Espero no haber olvidado a nadie. Pero muchas gracias a todos de nuevo por estar aquí. Thank you very much to everybody for being here. And we should have the reception. So you are all more than welcome. And yeah, thank you very much again. I forgot someone now. Andrea, you are watching, I hope, from Guayaquil. thank you for being family to me as well these years and for taking care of me so many times. Thank you.
So the real diploma is not yet printed, probably due to Corona, but I'm glad to have your certificate.
Thank you.
You have to sign that. Meneer met een dikke van het jaar.
Congratulations. Thank you very much. Thank you very much. Thank you very much.
I will not due to Corona, you know, but my brother has 5 people with Corona. So
Okay.
it's enough. Okay? Ah, we should a picture. Als jullie allemaal in not briefing, 1 minute, and you take the picture.
Quickly. John?
It seems apnea. Okay.
This is.
Thank you very much. Thank you very much. Thank you. Yeah.
But your husband is was too emotional. Thank you.
I got an offer from Jaime.
Yes.
So maybe I will join.
Very special, yes.
I look forward to that. Okay, congratulations.
And Wageningen.
When I had to speak Chinese,
Okay, perfect. Thank you. Are you gonna drink? Okay, okay. Both of you were always really I really appreciate it. Thank you very much.
Thank you very much. So kind. Wow.
One of the three of them. Oh, yeah. It's my hand. And this? For me? Yeah. I don't get to make it, but it's first as a requirement in the new forms, like for Greece, they said, okay, we should take care that there should be an equilibrium jury, but they don't tell how many female, but at least one female. Now you can relax.
I'm going to leave.
Sure.
¿Hay que llevar algo de aquí o te deja todo?